Good morning. Today we're going to learn a little bit about top-down proteomics. I've spent 20 years in the field of proteomics, and I've spent almost all of it on shotgun proteomics, or bottom-up proteomics, where we deal with the peptides we've digested out of proteins. But in this case, we're going to turn the table on its head and instead speak about top-down proteomics, the analysis of intact proteins. We're going to start with the rationale for why top-down proteomics, at least theoretically, offers some real advantages over shotgun proteomics. And alongside that, we're going to discuss a definition that's recently been put forward for proteoforms. So that's going to be a linchpin that explains why top-down proteomics has so much to offer. From there, we're going to spend a little bit of time talking about analytical chemistry. I know that might not be usual for a bioinformatics talk, but these are really key aspects of top-down proteomics that make it quite different from shotgun. So I think it's worth discussing in some detail. From there, we're going to talk about one of the largest identification suites out there for handling top-down data sets, and that is ProSight, or ProSight PTM, or ProSight Lite, their relatively new version. So discussing how that algorithm does its thing and explaining its relationship to bottom-up proteomics identification algorithms is, I think, very worthwhile. From there, we'll talk about a rather different kind of algorithm, one based on dynamic programming that uses an approach called spectral alignment. It's really become a very valuable technique for analyzing very complex post-translational modification sets in particular. And finally, we'll talk a little bit about a consortium that's formed to help construct top-down proteomics as a field, as a technology, and talk about some of their efforts towards standardizing the way that we talk about proteoforms. So it's always worthwhile, I think, to start with the complexity of the human genome. There are several different numbers that we throw around when we talk about how many products a human cell can make. I think it's worthwhile to kind of work our way through this. I've decided to use GenCode 34 as a basis for this discussion, but there are other annotations and so on that we could work from. This one was released in April of 2020, so it's relatively current from the time of filming. So we see here that they list the total number of genes as 60,669 in a human. And that number probably seems really high if you've heard me lecture a lot and talk about 20,000, 20,000, 20,000. But I'd point out that the number of protein coding genes is still under 20,000. It's 19,959 in this current release of GenCode. So what's the difference between this almost 61,000 and almost 20,000? The difference is that we have a large number of pseudogenes. Sometimes these reflect things like retrotransposons that have written themselves into the DNA, and long non-coding RNAs. There's quite a number of these. You can see that they account for about 40,000 of the genes that we have in our genomes. But of the stuff that actually produces protein-coding genes, of the genes that actually code for proteins, we see that that number is floating around 20,000 still. We see that there were 84,068 transcripts formed from these genes. Those transcripts the number of transcripts from protein-coding genes is quite a lot larger because we have the ability to splice together different exons from a gene in order to produce a different transcript from it. So when we look at the 84,000 sequences, we often discover that some of them produce the same protein product. If you were to translate these different transcripts, you would still come up with the same sequence. So although we have these 84,000 well-documented transcripts, we have only 62,000 distinct sequences after translation. And finally, we can then ask, of all the protein-coding genes, what fraction of them, or what number of them, have multiple distinct translations, which is to say you can get different polypeptides when you translate the transcripts produced by that gene. We see that that number is 13,717. Another way of saying it is that 69% of all human genes can produce multiple protein sequences through the process of different transcription. So that's a pretty shocking number if you think about it. It is normal for a human gene to have multiple translations available because of exon shuffling that produces these different transcripts. So if that's the case, we should expect that different isoforms different transcripts produced from the gene can be observed in different proteoforms. And that is one of the key things that causes this proteoform technology, proteoform definition, to be so useful to us. We can talk about the different isoforms, each corresponding to a different proteoform, which is to say that transcripts that translate differently produce different protein products in terms of their sequences. But this is not the only way that protein sequences, that different proteins may differ in form from another protein produced by the same gene. So we start with genetic variation, sure. We may have a different allelic variant that has a non-synonymous SNV that changes which amino acid is put into the polypeptide. That's one way that you can change the sequence. The RNA splicing may mean that you have a different transcript, a different isoform produced, and thus a different translation from it. It may also be, though, that different clipping can happen. I think lots of us know that very frequently the methionine at the N-terminus of most proteins gets lopped off. So that's one form of truncation of a protein sequence. But maybe we don't think so often about the expected proteolysis that takes place for from a signal peptide that appears at the N-terminus of that protein sequence. Once that protein is shipped to the proper location, that initial peptide, the signal peptide, can be truncated, and thus we have a different proteoform from when this protein was initially translated to when it actually appears on site where it's supposed to be. So that's certainly possible to truncate a protein sequence from the product made by the ribosome to the mature protein that's ready to take on function. And of course, post-translational modification is a tremendously important reason why a proteoform may differ. That a protein may be in a relatively inert state, but you slap a phosphorylation on it and suddenly, boom, it springs into action. This is a protein changing its proteoform because of the action of post-translational modification. If you haven't had the chance yet, we've recorded a lecture on post-translational modification identification. It's really canted at shotgun proteomics. So this lecture is going to spend more time on the issue of how we deal with finding PTMs in the context of an entire protein at once. Okay, so this diagram from the Lloyd Smith paper in 2013 helped to explain all these different sources of variation that go into a proteoform differing, where different gene products from an individual gene may differ from each other through sequence changes, through cleavage, and through post-translational modifications. All right, so let's try to ask the question from another light. If you are, like me, from the bottom-up proteomics world, you're probably accustomed to the way things are done. And the way things are done is that you cause your proteins to denature, you reduce the disulfide bridges and alkylate those, and then you hit the thing with trypsin and it cuts it up into a bunch of peptides. Our unit of measurement then is the peptide. The peptide comes into the mass spec, it's collided with gas, it blows apart, it produces fragment ions, we measure those in a tandem mass spectrum and we identify them. That's grand. But what's the shortcoming of this? Let's start with a gene called VEGF. VEGF is interesting to people who work in cancer particularly. If I remember the name correctly, it's vascular epithelial growth factor. I think a receptor for VEGF, basically, for VEG. So here we have two different sequences that have been posted to UniProt for this gene. So we see that the isoform VEGF 189 has this sequence, and the sequence for VEGF 206 is slightly different. Now, as I recall, VEGF 189 is normal. If you go hunting in cells for VEGF, this is probably the version you're going to see. Down at the bottom, we have a much rarer transcript that is found. I don't know if it's associated with cancer. I've just mocked up an example here, but it's clearly a different sequence, right? We see that that this region down here in orange differs between these two sequences. Now, I want you to remember that in shotgun proteomics, we're going to measure individual peptides drawn from the proteins that are present. So let's imagine that we have a peptide that starts at AEG here that runs through VVK. If you're squinting to look at the screen right now, you really should go to the description appearing below this video and download the PDF. It'll be a lot easier to read the slides from that, trust me. Alright, so you can imagine then that we have a peptide that stretches from here to here, another peptide that stretches through this really long region up to this K, and then another one that continues on from PSC, etc. And so all these blue regions represent measurements. They are peptides that we have observed from these proteins that are present. Now, you might think if you see a peptide in this orange region, which is the part that is specific to this isoform, that you can say, if I'm trying to evaluate if this isoform is present, I just need to measure a peptide in that differential region. But I note that this HLF peptide that we were so excited about just a moment ago also appears in this other isoform. In other words, we have an extra exon getting inserted into the sequence, but this HLF one, even though it's in this region of difference, is just moved later into the sequence. So HLF does not, in fact, differentiate these two isoforms. We do, however, have a peptide down here in CCLMP that is only found in VEGF206. So if you were to identify any of these peptides except for the CCL one, you would not be able to discern whether VEGF189 or VEGF206 was present. The only information you have that explains that 206 itself is present is this peptide that is specific to its sequence, is explainable only by its sequence. Which means if you were trying to make a judgment about differentiation, to say our special VEGF variant is highly upregulated in cancer samples and present at only very low levels in normal samples, you would only be able to make that judgment on the peptide specific to that isoform. This is an area where a lot of bioinformatic toolkits for understanding proteomics datasets really fall apart because we're generally focused on just trying to produce the shortest protein list possible to explain all the peptides we've seen. So that's a big challenge for us to be able to say which isoforms are changing in expression from a given gene. If you're looking at the protein in toto, if you're looking at the intact protein by top-down proteomics, that kind of ambiguity is not going to be a problem anymore. Okay, next up, post-translational modifications. When we work in shotgun proteomics, we frequently make a mistaken assumption, which is that each post-translational modification is independent in its presence or absence compared to all the others on that same protein. Lovely tea. Okay, so what we have then is a list of different post-translational modifications known to be associated with histone H3,3. Now, you might be very interested in epigenetics, or the epigenome, to understand which parts of the genome are actively being transcribed and translated, and which ones are not. Histones and their post-translational modification states are a really big deal here. So you can learn about that from this Gary et al. Paper in Nature Genetics. But at the moment, let's just think about each of these as a different mark on this histone. We have K4Me1, which is to say the lysine at position 4 in this protein sequence bears one methylation, okay, a plus 14 on just that individual K4 site. We also have K4Me3, which is to say a trimethylated lysine 4. Now, it should be very obvious that lysine 4 having two different PTM states here is already in a case where independence cannot be claimed. If this residue bears methyl 1, bears a single methylation, it does not also bear a triple methylation. It's one or the other, or it's just unmethylated altogether. So these two cannot coexist, and therefore they're not independent PTMs. Down here we have a lysine at position 9 that may be trimethylated, a lysine at position 27 trimethylated, lysine 27 acetylated. That's a tough one, right? Trimethylation and acetylation have nearly identical masses, so it's very difficult to tell them apart unless you have a very high resolution mass spectrometer. All right, and then K36 may be dimethylated or may be trimethylated. You could think of K4 and K9 and K27 and K36, all these different lysines that get modified in this histone sequence. You could think of each of these as independently being modified. But the reality is that post-translational modifications are frequently very correlated in their activity. If K4 is methylated, then K27 must be trimethylated. We're still working out the logic of this stuff. But to make the assumption that they are independent from each other is likely to be very, very mistaken and is likely to lead you very much awry. But what if you're measuring the presence of this K4 methylation by the initial peptide of the protein, and this peptide does not also contain K27? You may detect a modification on K27 from another peptide, but because it's another peptide, you don't know that this K27 modification and the K4 modification were on the same protein. This protein may have different proteoforms, one that's unmodified, one where K4 is modified, one where K27 is modified, and one where both are modified. The only way you're going to know that is if the spectrum you have contains both modifications. That's where top-down shines. But bottom-up shotgun proteomics is frequently going to miss out on the co-occurrence of post-translational modifications. Well, if we should be measuring intact proteins, why doesn't everybody do it that way? Why not start there? The answer is that analytical chemistry of intact proteins is frequently much more demanding than of individual peptides. I think this quote kind of has it all from Chen et al. In their 2018 paper. Numerically, the intact proteome appears to be a much simpler mixture than its corresponding peptide digests. I've said this before in my shotgun lectures. If you digest the proteins of a mixture, your problem gets harder in a lot of ways. You might have started with 10,000 proteins, and now you have 100,000 peptides. So numerically, it looks like going the shotgun route is more difficult. In practice, however, protein-level fractionation and separation are daunting tasks due to the diverse physicochemical properties such as size, charge, and hydrophobicity, and the wide dynamic range of the proteome. So if you are looking at a protein that sits mostly within a transmembrane region, you're going to find that the hydrophobicity of that is quite extreme compared to a protein that, wiggles about wild and free in solution. One is very polar, one is very hydrophobic. If you're trying to create an analytical chemistry approach for separating these uniformly, you're going to find that it's probably catered to in-solution proteins or to membrane proteins. That's a problem. So that's an example of a hydrophobicity difference. We're going to talk about the range in protein size in just a moment, but we can also think about charge. It may be that if you are looking at something like a histone, you have a tremendous amount of basicity associated with this protein, and that basicity translates to a very positive charge. If you're dealing with a separation of your proteins that discriminates against highly positively charged proteins, away they go. So we need to be able to deal with this great range of biochemistry as we work. You might look at this slide and say, I can't read that. Don't worry, you don't have to. I wanted to portray visually some of the proteins that we very frequently think about in proteomics and illustrate just how different they are in size. Now here I'm giving the sequences, so the sequence length is what's going to be the visual cue you're seeing, but I've also written in the masses of these in Daltons. So we start with our friend insulin. If you I believe insulin may have been the very first protein sequenced, unless I missed my guess. Was it myoglobin or was it insulin? Anyway, insulin is very, very short. Here we see that in just under 12,000 Daltons, we have this sequence running from here to here, just 2 lines of a FASTA file. It's a very, very small protein. From there we can go to something like hemoglobin subunit alpha. This is a 15.25 kilodalton protein, and we see that it takes up about 2.5 lines within our FASTA file. We can then step up to trypsin. Yes, we often think of getting trypsin from bovine or porcine sources, but humans also have trypsin genes, and here we see that trypsin 1 comes in at a mass of 26,500 Daltons. So not a huge protein for taking on that kind of complexity. And its sequence is just over 4 lines of FASTA sequence. When we get to something like actin, we're getting a little heftier. You can see that we're now up to about 42,000 Daltons. We see that the number of sequence lines it takes is a little more than 6 lines of sequence here. And then we get to something that kind of crosses over this threshold. And this is a threshold at present, but it may not reflect where we sit in 2 or 5 years. So for now, I want you to think of something like keratin type 2 cuticular as a bit of a boundary for us. It's stepped over 50,000 Daltons. 50 kDa is a pretty large protein. Now, is that an unusually large protein? Definitely not. But if you're in the top-down space, as soon as you step beyond 50,000 Daltons, you'll frequently find that your ability to recover a decent sequence from it has diminished. So this boundary is changing a little higher and higher every year, but with current technologies, being in the 50 to 100,000 Dalton range is problematic frequently. Okay, so here we see that keratin, our old friend, is not that much bigger than actin, but it is enough bigger that identifying its sequence from top down may be problematic. And then we step up to the very most popular protein for everyone working in human biofluids, serum albumin. Serum albumin comes in at about 69,000 Daltons, and you can see its sequence is a bit longer. Now, by this last one, you might be thinking, gosh, that's a massive protein. This is collagen alpha 1 from humans. Weighs in at just about 139,000 Daltons, so hefty, right? Is it the biggest protein in humans? Not even close. Not even close. If you look at something like human titin, you'll be looking at a really large sequence. If I try to put this on screen, it would basically have as many lines of sequences as everything here, plus more. It's a very big protein. So when we get into these larger and larger sequences, trying to make sense of the spectra they produce becomes more and more problematic. All right, so how do we deal with the separation problem here? If you are accustomed to shotgun proteomics, you're probably very accustomed to thinking of reversed phase liquid chromatography. And in most cases, when we are separating peptides, the beads we use for RPLC are covered in C18 resin. So we have beads that have these long hydrophobic chains, these C18 chains pointing off in all directions. So there's kind of like a fuzzy sphere, basically. And each of those chains has a high degree of hydrophobicity, which means that a peptide will stick to it. But even better, if we cause a rise in the hydrophobicity of solvent in which these peptides and beads are found, we find that at a high enough degree of hydrophobicity, we can knock the peptides off of those C18 chains. That is not so likely if you have a protein. Proteins have some sort of hydrophobic region on them somewhere generally, and it will form a relationship with the C18 material so strongly that that protein will not be removable from the bead. So you'd better have something much shorter to work with. And frequently we find that even just a 4-carbon chain forming the lawn on this little sphere, this little bead, is about right in hydrophobicity for us to capture the protein, but be able to release it through a hydrophobicity gradient. Okay, now I hate to say this, but I'm going to include 3 equations in this talk, and they're worth looking at just because they help explain the relationship between things when we read papers about separations and top-down. So what we're looking at is the relationship between the pressure of the pump you're using to drive this hydrophobicity gradient, the viscosity, which is to say, kind of how snotty is the fluid you're trying to push through the column, the velocity, the rapidity with which you are moving things through the column. You can think of velocity as flow rate here. And generally speaking, our best sensitivity in mass spectrometry comes with a very low flow rate. And the length of the column. You might be accustomed to thinking of packing material of maybe 5 centimeters in a column that you use for proteomics. In top-down proteomics, we see people using columns as long as 1 meter. So that is quite a big factor. And you can see that the amount of pressure required rises as a linear factor. If you go from 5 centimeters to 1 meter, you have 20 times the length factor, which means that the pressure required is 20 times higher to drive that column. And finally, particle diameter. The beads on which we find these hydrophobic chains can be very large or they can be very small. And basically, the finer the beads are that you pack into your columns, the smaller the denominator, and thus the larger the pressure required. So smaller beads imply a higher pressure to drive the column. These things are worth knowing about. This kind of relationship is worth understanding as you read the papers about separations in top-down proteomics. And we have to talk about fragmentation for just a moment here. In tandem mass spectrometry of peptides, we spend a lot of time talking about CID, or its cousin, HCD. So CID is collision-induced dissociation. HCD is a slightly higher energy variant of that that's typically used in beam type rather than trap type collision-induced dissociation. So they're not that different. In this case, though, we really need to talk about electron transfer dissociation, which is a really important technique when you're dealing with very large, very highly charged protein ions. I would remind us of Ropsdorf and Fullman. I found this diagram on Wikimedia Commons. Typically, we would put the B ions on top and the Y ions on bottom, but here they've flipped that upside down, but we'll forgive them this. So we can see that for a given amino acid, you can always find the amino acid by remembering the ship number for the USS Enterprise, NCC. Okay, so here we go, NCC, NCC. If you can find NCC, you can find individual amino acids in a backbone, so NCC. Right, normally when we break a peptide bond through CID, we generate a Y ion and a B ion. That's the breakage between successive amino acids at the peptide bond. But in this case, we're going to be talking about an alternative fragmentation that does not break peptide bonds, but rather the bond between the nitrogen and the alpha carbon. So we'll talk about that in just a moment. Okay, so in collision-induced dissociation, we have a bunch of peptides in orbit, basically. And inside the trap, these peptides are orbiting along with a bunch of noble gas molecules in there. So we see them just traveling around. Maybe they're taking on kind of a complex saddle shape, depending on how this trap is set up. So we ordinarily we find that these collisions with gas kind of slow them down and kind of ensure that we can trap ions very efficiently in this ion trap mass analyzer. After a while, though, when we decide that we have isolated away to just the set of peptide ions corresponding to a particular mass to charge, that we can tickle them. We can give them a little extra energy, causing them to orbit even more rapidly around in the CID. In the trap. That means that the number of collisions they're undergoing in every second rises dramatically. And now, instead of kind of cushioning them and keeping them all in some sort of stable orbit, these fragmentations are these impacts with gas are causing the proton sitting on these peptides to start moving around. That creates a dangerous situation because eventually it sits down in a place where it destabilizes the peptide bond, causing this cyclization reaction, and you get a B ion and a Y ion. So it is an impactful process, and that has its pluses in that pretty much any peptide will fragment if you throw enough energy at it, but it has the disadvantage that in some cases these impacts can be very disruptive and very non-random, meaning that they can knock off post-translational modifications you would rather study. This has been a big problem for people who study things like Oblichneck. But it can also have the problem that you get very directed fragmentation to just a few sites, and thus a not very informative tandem mass spectrum. Now, Josh Kuhn and Saika and a whole bunch of other great folks put together a really cool technology that circumvents a lot of these problems for collision-induced dissociation. You might think that if that bashing this peptide with a whole bunch of impacts until it falls apart is a bit of a disruptive process, and you'd be correct. So electron transfer dissociation is intended to be a very gentle process by contrast. So in this case, the gas molecules are not really playing much of a role at all. What we have are a bunch of charge carriers, of anions being brought into this to do an ion-ion reaction within the mass spec. So we have a bunch of copies of our proteins, let's say. They're pretty heavily positively charged because we've added charge through the pH and the electrospray apparatus. But these charge carriers are negatively charged. Obviously, if you put a bunch of negatively charged ions in alongside a bunch of very positively charged ions, you're going to see reactions between them. And you might think to yourself, well, that's a neutralizing reaction, why would I care? But I want you to keep in mind that these anions are typically coming in with a single negative charge, and these proteins are coming in with multiple positive charges. So what happens? This is a part where you'd better talk to an analytical chemist to get everything right, but I'll try to give you the hand-wavy version. That we have an electron that's being transferred to a peptide ion that's carrying multiple charges. So we see that here we have a positively charged lysine in interaction with a carbonyl. This electron shows up, and yes, they neutralize, but they also have now created a radical. You see this little dot here, that's our symbol for that. This radical position can transfer around until it eventually sits at this, what was previously a carbonyl. And once this happens, we find that a breakage between the nitrogen and the alpha carbon has taken place, where the alpha carbon is now the radical. So this is a relatively gentle process. It doesn't require multiple collisions. It requires one chemical reaction between a singly negatively charged carrier and our multiply charged positive protein ion. So we drop one charge, create a radical, and now the protein has split. So its charge, which just dropped by one, is now divided between the two ions that depart. The structure for this breakage between an N and its alpha carbon and inside the amino acid creates what's called a C ion and a Z ion. And one of those is a radical as well to reflect the mismatch in number of electrons. So what we've produced then is a relatively gentle way to break apart a multiply positively charged ion, dropping its charge by one, and breaking it between the N and C of a particular amino acid. Just as you can have a ladder of B ions or a ladder of Y ions, you can also have a ladder of C ions and a ladder of Z ions. That's an ETD tandem mass spectrum. Very popular technique, especially in light of top-down proteomics. This brings us to our intermission, so take a quick moment and we'll be right back. And welcome back. In this part, we're going to move from thinking about analytical chemistry to the algorithms we use for making sense of top-down tandem mass spectra. We start with something that's not, strictly speaking, identification, but is quite necessary to making sense of a lot of the spectra that we deal with. I want to talk about two ways that a particular fragment may appear in multiple copies throughout the spectrum. Imagine that you have a C15 ion. This represents the N-terminal fragment produced by ETD. That C15 means that there are 15 amino acids in this fragment. It's quite likely that when you have 15 amino acids, that you have at least one extra neutron sitting in one of those nuclei, which means that you're going to get the monoisotope plus a C13 peak, you might say. Or you might have two extra neutrons sitting in those nuclei, in which case you'd have a monoisotope plus 2 peak. These form what are called isotopic envelopes. So if you have a particular ion of a given composition, it's likely that you see an isotopic envelope for it. The second case is that when you have an ion with 15 amino acids in it, the probability that it takes on a single charge is stacked next to a probability that it takes on a double charge instead, or a treble charge if you've started with something that's charged highly enough. As a result, it may be that you have isotopic packets for the +1 version, and the +2 version, and the +3 version. Now these don't have the same chemical composition because they have different numbers of protons on board. But in this case, a single ion may appear in multiple peaks due to isotopic differences, and it may appear that you may have different isotopic packets observed at different charge states as well. That means that there's an awful lot of duplicity in this tandem mass spectrum. We often find that it's very valuable to move all of this current spread out among all these different isotopes and all these different charge states back into one peak to give us greater certitude that this mass to charge value for this monoisotope is at single charge or neutral charge represents all of those peaks that were sprayed across the spectrum. So deconvolution is the approach that we most frequently use for this. Here you can see a particular protein that's charged pretty highly. And how can you tell? Well, look at the x-axis on this. This is M over Z 3122, M over Z 3123, M over Z 3124. So within one M over Z, we've got this whole set of isotopes seen at a given charge state. You can infer what charge it is by the spacing. So if in the space of 1 M over Z you have 5 isotopes, that's a pretty good indicator you have a +5 ion that you're observing. So by being able to recognize these isotopic envelopes and further recognize that multiple isotopic envelopes correspond to the same mass to charge value, gives us the ability to represent all of these peaks at different charges with one in a de-isotoped, deconvoluted spectrum. This is a frequent first step in the software out there. One of the first approaches to this was created by David Horn, and it's called Thrash. What I'm showing in this case is one from a researcher associated with the University of California, San Diego, Xiaowen Liu, who created a software specifically for fragmentation spectra from top-down ions, from intact proteins. All right, so that's great. We now have some notion of how to go about this. Now, if we were just adapting a database search engine for the purpose of identifying tandem mass spectra, we would simply need to allow for the software to accommodate different truncations of sequence, for example, removing signal peptides, removing initial methionine, stuff like that, and allowing for PTMs to be scattered across the amino acid sequence. Let's imagine that the only PTM we need to take into consideration is methionine. Methionine is a pretty rare amino acid, and yet if you have a protein sequence that's 200 amino acids in length, the odds are you're going to have multiple methionines on board. As a result, you probably need to take into consideration the possibility that you have one methionine oxidized, 2 methionines oxidized, 3 methionines oxidized, and by the time you get up to the content of 5 or 6 methionines, the different patterns you have to consider just for the possibilities where 3 of them are oxidized becomes quite complex. So you can see that we have this huge expansion in the number of potential proteoform spectrum matches that we must consider. Now, you might have read this very quickly and said, he's talking about PSMs. We know all about that from shotgun proteomics. But when we do top-down proteomics, we're no longer talking about a peptide sequence match. We're now talking about a proteoform spectrum match. So we often see that abbreviated PRSMs. Okay, so as in shotgun PTM identification, we're going to decorate the peptide, or in this case protein sequence, with allowable mass shifts. Top-down benefits from known PTM annotation. In database search for FASTA sequence databases, most of our tools like Sequest and so on typically say any methionine may be oxidized. And if you set up for a phospho search, any serine, any threonine, any tyrosine may be phosphorylated. That's great. But in fact, we have a fair bit of knowledge about known post-translational modifications on these sequences. They just don't appear in our FASTA sequence databases. So it might be that you're making use of a PEF-formatted sequence database. PEFF is a format created by HupoPSI to pass information like sequence variants and PTMs on the description line of what looks like otherwise a FASTA sequence database. It might be that you're searching against a Uniprot database that you've downloaded in something other than FASTA format. In that case, known PTMs may be available along with the sequence data. When we get into top-down search for PTMs, we very frequently benefit from extensive annotation of known PTMs as a basis for our understanding the spectra. Okay, frequently, although sequence tagging has not really taken off as a standard technology for interpreting tandem mass spectra of peptides, it's frequently used in the context of protein tandem mass spectrometry. The reason is that with 50 amino acids rather than 20, you have a much better shot that somewhere in the spectrum you have a series of consecutive fragment ions that you can interpret sequence for. So having the ability to infer sequence tags out of the tandem mass spectrum helps us to cut down the search space within the protein sequence database rather significantly. And finally, if you're using a protein sequence database with annotation, like the Uniprot downloads that are not in FASTA format, you probably have some indication of where signal peptides are believed to be on these protein sequences. And so in silico, you can think about what would the sequence look like if we remove the signal peptide. Very helpful in this case. All right, so ProSight PTM is one of the most widely used, best marketed, good training, long-standing development toolkits out there for top-down proteomics. It's an impressive piece of software. It is not the only one we're going to talk about today, but it's one you should definitely know about because in a lot of ways the toolkits for top-down proteomics that are most mature, those that have had the longest to marinate are the ProSight toolkits. So I wanted to talk about its three-way search assessment ability. So let us imagine that we have a sequence database that represents known cleaved forms of proteins. So here that's represented by the ProSight warehouse, a SQL database. We have mass spectra that show us very accurately what the mass and charge of a particular protein ion is, or proteoform ion is. So the intact ion can be played against the ProSight warehouse to come to undergo a process of selection by mass, to say that only those proteoforms for which we have observed intact masses as precursors will be considered for comparison to the resulting tandem mass spectrum. So that's one route, the absolute mass route for interpreting this tandem mass spectrum and matching it to a proteoform on the basis of what is very similar to the precursor mass filter in database search. So green gets us there. You could think about this from the opposite end, though. What if instead we looked at the observed fragment ions, C's and Z's in the case of ETD, or B's and Y's in the case of HCD or CID, and then we can say, can we find tags of sequence from the tandem mass spectrum to say, we have these 5 amino acids in a row. What protein sequences contain those 5 amino acids in a row? Go against your FASTA file, and now you have candidate proteins that you can score. And of course, it may be a case that you're able to combine both the information about intact masses and fragments, particularly with a tag, for scoring things. Now, consider the possibility that you are able to narrow down a very small number of protein sequences based on intact mass of the proteoform and on the basis of the fragment ions you've observed, you got this tag. So we have a relatively small number of proteins now to compare to a tandem mass spectrum. But how do we handle the fact that there may yet be some ambiguity about which particular proteoform? For example, you may have said, I know this protein plus 3 phosphorylations and 1 acetylation equal the mass that I saw for the intact ion of the proteoform. And I've confirmed internally that the sequence matches to this FASTA sequence. That's great. But I don't know where this 1 acetylation and these 3 phosphorylations are located on the sequence. For that, you're likely to go through an expansion process to say, what are all the possible decorations of this set of sequences that allow us to put 3 phosphorylations and 1 acetylation in place? And then you need to be able to score those so that you can rank the best proteoform to explain all the fragments you've seen at the top, and put the ones that are the least likely interpretations by that score at the bottom. How do you do that? For this, we're going to use an approach that's very similar to what we've frequently seen in database search. In particular, 3 years after this was published, OMSA, the the Open Mass Spectrometry Search Algorithm from Louis Gehr et al. At NCBI was published. They used almost the same kind of math. Both of them use a Poisson distribution-based system for scoring the number of matches. Very frequently we see that match counters are one of the most common ways that people evaluate the quality of a PRSM, or for that matter, the quality of a PSM, of peptide spectrum match. The notion being, if you have some protein, you can consider all the fragments that should be produced by it, and then ask, which of these fragments do I have experimental evidence of in my tandem mass spectrum? Okay, so we have our last 2 equations for today. I hope you'll tolerate this for just a moment. What we're trying to compute is P, the probability that by random chance we would have a match this good for this sequence and this spectrum. Okay? So we have X, F, N, E, X, F, and N here, all showing up at the top. I've tried to add definitions of each of these. So E, I hope people remember, is a value of like 2.8128, I think. It is the base that we use for natural logarithms. So that's just 2.8. Substitute that in. All right. Now, we have X, which is the mean probability of random match. It's going to get defined in its own equation. So hold X aside for just a moment. We're going to come back to that. We have F. F is the number of predicted ions. Obviously, if you have a very long sequence, it's going to produce a much larger number of expected fragments than a very short sequence would. Ideally, and well, typically, we expect that a given peptide breakpoint will produce 2 fragment ions in the tandem mass spectrum. So, for a given number of predicted fragment ions, we can ask how many did we actually match? So what you see then is that matching 5 fragments rather than 6 fragments produces a big drop in this denominator. That if you have 5 factorial and 6 factorial, it's really easy to remember that 6 factorial is 6 times as large as 5 factorial. 7 factorial is 7 times as large as 6 factorial. So matching even a small number of additional fragment ions makes this denominator big, thus giving you a probability of random match that's much, much smaller. That's an important thing to keep in mind. But the more fragment ions that you predict, the larger, you have this change in the numerator. So expecting fewer fragment ions and matching more of them is going to drive this probability down. And a low probability here is the signal that this is a good PRSM. Now I said we were going to come back to X. This is our probability of a random match. So we need to know how busy the tandem mass spectrum is. How densely packed do we expect the fragments to be along the x-axis? So we take into account the frequency-weighted average amino acid mass, which is about 111.1 Daltons. We know that each peptide bond that breaks has 2 fragments that it produces. And we have a mass tolerance. So ideally, if you're dealing with tandem mass spectra of proteins, you definitely want to have a very high resolution, very mass accurate instrument. And we're going to allow for some amount of leeway on either side of our predicted location for a fragment ion where it can be found. So this is very similar to the fragment mass tolerance we use in shotgun proteomics. So we have this expected location, we have some amount of error allowed on the downside, and some amount of error allowed on the upside, so we have to double that. So this math all comes together to produce a single probability that this particular match happened only by random chance. So how can we visualize something like that? It's kind of complex, isn't it? Well, because of Ropsdorf and Folman, we have this common hook visualization that we can use to explain whether or not we had evidence for a particular fragment ion from a protein sequence. So here we're looking at a fairly long one. We're looking at something like 110 amino acids, I guess. And you see that some of these locations have little hooks on the upside. You see that? So the top part is hooked to the left. Here we have one where the bottom hook points off to the right. So this is, if these were CID data, I don't remember whether this is CID or ETD, but let's just say it's CID for the moment. Then these hooks to the left would represent places where we saw B ions. In these cases where the hook goes off to the right, we say, that's a Y ion. Now it might be that we have an ETD spectrum, in which case this hook to the left would represent the C ion, and the hook going off to the right would represent, sorry, a Z ion, a CZ pair, right? So these visualizations are simply telling you, do you have evidence for this breakpoint within the protein sequence? If you see a protein sequence that's covered with indicators of N-terminal and C-terminal ions, you feel good about it. If you see one that has just a very few places marked out, you should probably be much more cautious about accepting that PRSM. The other thing is that you may be very interested in knowing whether a particular part of this protein is decorated. So let us imagine that you wanted to know, is the serine carrying a phosphorylation? There are no fragment ions next to that serine to give you information about that. So being in a region that's deserted by, that's devoid of fragment evidence means that you're going to have a lot harder time making any assertion about this particular part of the sequence. But I think everybody should be able to read these. Basically, if you see lots of ions in evidence, that's very good. If you see that all of the evidence comes from the N-terminus, or all of the evidence comes from the C-terminus, you should worry about that. We like to have a mix of N-terminal and C-terminal ions supporting these sequences. Alright, so that's a fair bit about ProSight PTM and ProSight Lite, but I would like to spend a fair bit of time talking about dynamic programming. We've encountered this before, particularly in light of sequence alignment. One of the most widely used sequence alignment algorithms out there is Smith-Waterman. Smith-Waterman is an example of an algorithm that uses dynamic programming to find the optimal match between a pair of sequences for local features. So in the case of Smith-Waterman, we have a sequence A and a sequence B. We find that there are locations where we can score it positively to say that, the letter here is the same as the letter there. Or we might use BLOSSOM, where we say this amino acid here is an acceptable match for this amino acid there, like leucine versus valine. But let's just consider the possibility of counting exact matches between these two nucleotide sequences. So this G could match that G. This T could match this T. This A could match this A. But we see that there are enough differences that it's not an obvious sequence alignment across the board. So dynamic programming allows us to find the optimal match given our scoring rules, in this case maximizing the number of identical amino acids between two sequences. Basically by filling in this table once, we are able to find out what the optimal alignment of one sequence versus the other is. Now it might be that over the time period of evolution that we've had some insertions and deletions. We note that this sequence is 1, 2, 3, 4, 5, 6, 7, 8, 9 letters long. This one is 1, 2, 3, 4, 5, 6, 7, 8 letters long. So we already have a difference of one letter between the two. It might be that a gap is required to explain the best alignment between these two sequences. So when we look at the best path for this, we see that it comes straight from the original position up through nucleotide 3, and then there's the shift to the right. This represents a gap that's been inserted in one of these sequences in order to cause a match to the other one. Alright, so this approach is great anytime you have this additive kind of optimization problem. Here we're just scoring +1 every time we can compel two letters to fit, but we probably also have some sort of penalty that we apply to introducing a gap. How does any of this have to do with proteomics, though? I thought it would be worthwhile to talk about a few examples of dynamic programming as used in shotgun proteomics, because it's actually pretty ubiquitous. So let's start with inferring sequences from tandem mass spectra. Some of the software that I was very cognizant of because of my PhD project was the work coming from Vlado Dancik and Pavel Pevsner, who wrote some really cool software for inferring the sequence directly from a tandem mass spectrum with no reference whatsoever to a FASTA database. This approach is very, very frequently used. It employs dynamic programming as a way to optimize which path you're going to pull out of the data. And it doesn't have to be software from these guys to do that. Most of the de novo tools out there will use dynamic programming as a way to pull the optimal path out of the relationships found between peaks. Aligning liquid chromatography retention times. If you have a pair of LC-MS-MS experiments and they have a certain set of features observed on the M/Z plane at a particular moment in time for LC, you're going to see some amount of deviation in what retention times are observed for a particular peptide. You know that making the smallest scrunches and extensions possible in the LC retention times to force a fit between them is best. You don't want to have to, invoke a 5-minute lag here and then throw in a 5-minute compression here in order to force things to fit. You'd much prefer that those differences are on the order of seconds. So here the paper from Ono et al. In 2006 demonstrated that this kind of approach for mapping retention times among raw files, among LC-MS-MS data sets, can be accomplished through dynamic programming. Certainly, localization is a very big deal when we're dealing with post-translational modifications, and even in shotgun datasets, we see that people have shown ways to apply dynamic programming to find the optimal location, the optimal localization of these modifications given a peptide sequence. That was published just in 2012. And recently, Bill Nopel's group, J.J. Haubert, published this article on finding the way to translate a cross-correlation score for this particular peptide sequence, or for that matter, all possible peptide sequences for a given spectrum, to translate the distribution of X-core scores into p-values using dynamic programming. So obviously this technique is very widely applicable. Now there's a great paper from 2008 from Ari Frank et al. This is yet another mention for the Center for Computational Mass Spectrometry at University of California, San Diego. Just one of their papers to show how to make sense of these data. So they found that there are a lot of issues that come into play when PTMs come into combination with proteoforms, what we now call proteoforms. That term didn't exist back then. So the candidate expansion method, this is the case where we know that a small set of sequences along with PTMs accompanying them that could be used to explain a PRSN. The candidate expansion method leads to an exponential growth in the number of candidate protein forms that need to be considered. When a computer scientist says exponential growth, they're concerned about something. So the number of possibilities we have to take into account is worrisome here. Top-down spectral alignment may deal with as many as 10 to 20 post-translational modifications for a protein. That's obviously a big problem. If you have 50 modifiable sites and 20 PTMs you need to distribute across them, you have a huge number of possible localizations that you need to take into account. Doing this by just expanding it out is not very viable. One often deals with multiple isobaric protein forms in the same spectrum. Now this is kind of rare because you don't generally see computer scientists calling out analytical chemists for shortcomings of their data. But sometimes the shoe fits. In this case, they're making the argument that multiple proteoforms of the same sequence may be visible with just different localizations of the PTMs. What that means is that an individual tandem mass spectrum may represent the data from multiple proteoforms that have co-fragmented. They have the same M/Z, they have a very, very similar retention times. As a result, one tandem mass spectrum contains fragments from multiple proteoforms. If you've got a situation like that, the software is going to be wrong if it chooses either of those as the answer, because there's the other answer embedded in the same spectrum. Okay, So when we look at dynamic programming in this light, the software is trying to find the optimal way to account for the mass difference between what was observed and what the sequence database says should be there by divvying it up into different post-translational modifications. So in this diagram, we're looking at a relationship between a sequence and the masses that are observed. So here's our sequence from the database. Here's the M over Z we're talking about. And now we are able to find ways to accommodate a shift between of 42 Daltons between all the fragment ions and what the peptide sequence should say there by saying, hey, the N-terminus is acetylated. You understand that this would mean all of the N-terminal ions have a mass that's apparently 42 Daltons higher than what they should be based on sequence alone. So the software is able to pick that up and also deal with the fact that there are 2 other acetylations that appear in just these first 15 amino acids. This protein is much longer than 15 amino acids. We're just showing the first part of the protein sequence. So we see that this diagonal is going to rise by 42 mass units to reflect this N-terminal modification. Then it can hit the fragment ions that are observed along the way. You see that there's this ambiguity here that K5, lysine at position 5, and K8, the lysine at position 8, are each observed in methylated form. I'm sorry, acetylated form, my bad. That was plus 42. Yes, so we see that it's either acetylated or trimethylated, either way around. And it may be that the spectrum has evidence for both variants, where the K5 is methylated in one case or the K8 is methylated in the other, or acetylated, sorry. But all of the spectral evidence agrees very strongly that K12 is acetylated for every copy of the ion that was observed in this tandem mass spectrum. So here you can imagine the software is trying to find the best way to add expansions within the sequence to reflect the added mass associated with PTMs. It has some total mass that the sequence mismatches it in. Here we had 3 acetylations, so that's going to be 3 times 42. So 126 Daltons differ between the first 15 amino acids and what the spectrum has to show. The software is now trying to divvy up that 126 Daltons into recognizable PTMs along with locations on that sequence. It's pretty impressive, but this approach is highly efficient compared to the massive expansion of all possible localizations for those masses. Now, this approach has become rather ubiquitous in new algorithms introduced in this purpose. So we started with a paper from Ari Frank in 2008, which introduced MS Top-Down. This is from the Center for Computational Mass Spec at UCSD. We saw that 4 years later, the algorithm was improved again by a fellow named Xiaowen Liu, who's going to show up in just a moment here. So he created some software called MS-Align Plus while he was postdocing with UCSD. That software, in turn, has gone on to inspire a lot of other code. Now, I note that the folks who graduate from the Center for Computational Mass Spec frequently went on to work in some very high-powered labs, and they took the best ideas with them to go on and continue expanding them into the future. So we see that folks from CCMS went on to work at Pacific Northwest National Laboratory, where they created the MSPathFinder. You can see the paper for it was released in 2017. They found a way to treat the different nodes of the dynamic programming graph as chemical compositions, which they found to be very useful in limiting the search space down. We also saw that the fellow who created MS-Align Plus, Xiaowen Liu, left from UCSD to become a professor out at a university we call UIPUI, which is short for Indiana University and Purdue University in Indianapolis, which is why we say UIPUI. So he went on to create an algorithm called TopPIC while he was out there that expanded massively on what MS-Align Plus could do. And more recently, they've worked on a technique using something called a mass graph that allows them to implement this dynamic programming algorithm very efficiently, even when large numbers of post-translational modifications are in play. So TopMG is, I'm not sure how that's pronounced yet, TopMG? I don't think so. So TopPIC and TopMG are both examples of software derived from this idea that they developed originally at the University of California, San Diego, now at UiPui. And the software is amazingly freely available for interpreting your spectra. MSPathFinder from PNNL is another example of people moving from UCSD to Pacific Northwest National Labs. This third link, however, is not an example of actual movement of people. It's a case where the idea has transitioned across the ocean. The folks who wrote pTOP are the same as the team that's behind the pFind algorithm that was created out at Beijing. So they're at the Institute for Computing Technology. It's a very mature suite of tools. pTOP is a relative latecomer. pTOP 1.0 was published in 2016. I believe they're on the verge of publishing version 2.0 now, which has added a lot of features on top of what was already there. But just by looking at the diagrams, it should be pretty apparent that that both of these routes are handled via dynamic programming. That this has become an idea that has come to be dominant among the computer science heavy groups that focus on the interpretation of tandem mass spectrometry. So I'm really excited to see so many different options have evolved now in the space of tandem mass spectrometry for top-down. Now for this last part, I want to spend a little time coming to some slides that were provided by my friend Richard Leduc up at Northwestern University. If you have a profusion of new tools in a space, it's not very helpful if each of them have a completely different format for describing which PRSMs they have observed, which proteoforms they have observed. So the group that Richard Leduc has headed has created this approach called ProForma, which represents a way to communicate to others in the press a standard set of proteoform definitions that relate to the data at hand. So how can we express amino acid sequences and the ambiguity associated with some of those? How can we handle an amount of mass missing at the N-terminal side or the C-terminal side of a sequence? How do we name the modifications within the sequence? What are the acceptable ways to describe an accession number? Now this is not a HUPO-PSI standard, but Richard Leduc has been coming to the HUPO-PSI meetings. I hope that someday this will become part of what we, how our group communicates proteoforms. But you can see that there's been a lot of thought here about how we can make the results from pTOP and from MS-Align and from for that matter ProSight, how we can make these all very, very comparable down to the level of PRSM to say this spectrum relates to this proteoform. We can match the sequence here and here to say these agree in their assessment of what the spectrum represents. In practice, ProForma ends up having has a relatively human-readable capability, which I think is very nice. When we think about the community file formats we use for communicating tandem mass spectra or PSMs in shotgun proteomics, we often find that they're not easy to interpret for a human just looking at the file. But that's really been an emphasis in ProForma. So I think that most people working on the bench would know that if they read GRGK square brackets acetyl along with a unimod code, they can say, there's an acetylation at this point in the sequence, and the letter before it was K. Oh, maybe the acetylation sits on lysine. So that's lovely. This is an example then of how to communicate proteoform information in an open format. I have a lot of heroes in my mind when I think about top-down proteomics. This area has been one that has struggled with, at first, the mass spectrometers not being good enough, and then the separations not being good enough, and then the informatics were not good enough. And rather than say, well, I guess I'm just going to work in a different field then, these are folks who've really plugged away at making these technologies work over time. So I think there's a lot of credit to particularly the Consortium for Top-Down Proteomics that has put together a lot of work. I've named just a few people off of their board of directors, and I mean no affront to people who didn't make it onto the slide here. But I think that if you've been to an ASMS meeting, you've probably had some sort of encounter with Neil Kelleher. I can say this because the man is made of energy. So Neil Kelleher has been throwing his energy into top-down proteomics for about as long as I've been a PhD. Actually, I think he started a little before that even. He's an incredible fellow, and if you don't feel excitement about the future of top-down proteomics and even about the present of top-down proteomics after talking with Neil Kelleher, you're just not very responsive to emotion and drive. He's a fascinating fellow. So big credits to him. I've mentioned Pavel Pevsner before in light of de novo sequencing in particular. He has also had a strong tie to the fine world of creating a home for things like MS-Align to be designed. I particularly give credit to Xiaowen Liu, who appears here at the bottom right. Xiaowen is not one of these noisy personalities out there. He's quiet and thoughtful, and I find that when I want to know what these tandem mass spectra represent for an intact protein, his software is what I always turn to. The man's really smart and has produced some very fascinating software. It is, however, friendly to computer scientists. You don't often find that the software can be run on anything but the command line. So it's really lovely to see that a team at the University of Wisconsin has published something called MASH Suite, which allows you to run tools from his lab through a much more intuitive user interface. Really glad to see that pairing is coming together. So as these tools gain in maturity and as they gain in usability for researchers on the bench, I think that we're really poised for an exciting place where top-down proteomics is practicable anywhere. There are lots of pieces to this talk, so I'm going to try to walk through some of the biggest points. If we digest proteins to measure them, we lose peptide-peptide relationships. That is a really big loss, and just because this is the way things have always been done does not mean that this is how we'll do things 20 years from now. It may be that top-down is ready for the breakthrough where ordinary labs would opt to go this direction. Top-down proteomics relies upon high-resolution MS-MS, and it relies upon good separations and fragmentation technologies. This has been slow in some places, that we don't see, for example, a lot of electron transfer dissociation capable instruments in laboratories here in South Africa, for example. So my hope is that over time, the availability of the separation technologies and improved fragmentation technologies will go hand in hand with high-resolution mass spectrometry, even here in South Africa. The dominant ProSight PTM framework has competition from alignment-based software. I don't think Neil Kelleher is going to mind my saying that he's always got to watch his back. The software of ProSight PTM is wildly impressive, and it's very friendly, and it offers very good support from the companies that market it. So that's lovely. However, it's getting easier and easier to use these spectral alignment-based technologies, mass graph alignment-based technologies. I think that those techniques have serious advantages as we get to larger and larger proteins, being able to characterize them in top-down. The Consortium for Top-Down Proteomics is impressive. They've put together professors from a good number of schools that are making these technologies more accessible. They will not be content until any lab anywhere in the world can make use of these technologies, and I'm really grateful. We should have people like this pushing us to take on new technologies. Who knows what the future looks like, but I hope that this talk has given you a brief intro into what top-down has to offer.